welcome to everyone who's just logging on for today's SSR webinar. We're just going to give it about a minute here for folks to get connected. Welcome to everybody who's logging on and joining us. We're just giving it a few seconds for folks to get connected here. All right, well, hello everyone and welcome to today's SSR webinar. If you have any questions about this webinar, please feel free to reach out to us directly at SSR headquarters via email at membership@ssr.org. This webinar is scheduled for 60 minutes and it is being recorded. It will be posted on the SSR website shortly thereafter for members to access at their convenience. In just a moment, our moderator will begin with a brief introduction followed by our speakers' presentations. Please feel free to submit your questions during the presentation using the Q&A text box on the bottom middle of your screen, and our moderator will read them to the speakers when the question portion of the webinar begins. With that, I will turn things over to Michaela.
Welcome to the SSR webinar on gene editing in domestic animals. I'm Michela Ciccarelli, an assistant professor in comparative telegenology at Washington State University, and I will be your moderator today. I'm very excited that you are so many joining us today because we have two pioneers in this innovative technology as our speakers. Their expertise and experience in this field will provide valuable insight and knowledge to you. As we said, we have time for questions at the end, but please feel free to send them through the Q&A feature at any time. Our first speaker is Dr. John Audley. Dr. Audley is the Associate Dean for Research and Director of the Functional Genomics Initiative and Center for Reproductive Biology, and is a tenured professor at Washington State University. His research focuses on engineered food animals with enhanced resiliency and production efficiency traits using gene editing technology and devising reproductive biotechnology to effectively disseminate heritable genetic modification. Dr. Audley has published more than 90 scholarly works in this area of research and has been awarded multiple grants from the National Institute of Health and USDA National Institute of Food and Agriculture. Recently, Dr. Audley became the first academic scientist to be granted investigational food use authorization by the Food and Drug Administration to introduce edible product from CRISPR-Cas9 edited livestock into the human food supply. Beyond research, Dr. Audley has worked with the FDA, American Association of Veterinary Medical Colleges, Association of Public and Land-Grant University, and National Academies of Science in workshop and task forces to help inform policies on gene editing of food animals. He also testified before the United States Congress on the 21st Century Biotechnology application for animal agriculture. Dr. Audley received his Bachelor of Science from the University of Nevada, Reno, Master and PhD degree from Washington State University, and completed a postdoctoral fellowship at the University of Pennsylvania. I'm also honored to introduce our other speaker, Dr. Mike McGrew. Dr. McGrew is a professor at the Rosalind Institute, part of the Royal Dick School of Veterinary Studies at the University of Edinburgh, United Kingdom. Dr. McGrew research at the Rosalind Institute study a special stem cell, the germ cells, which produce sperm and eggs. These cells can be used to generate genome-edited chickens, which contain precise genetic changes in their genome. They use this system to investigate susceptibility to avian influenza infection, sex crewing in chicken, and novel way to preserve both chicken breeds and avian wildlife. Dr. McGrew research has been cited widely in several popular scientific books, such as Tamed by Alice Roberts, How to Clone a Mammoth by Beth Shapiro, and Bring Back the King by Ellen Pritchard. Dr. McGrew earned a Bachelor of Science from the University of Minnesota, a PhD in biochemistry from Boston University Medical School. He completed postdoctoral training at the Institute of Developmental Biology of Marseille in France, and the Rosalind Institute. He was recently awarded a chair in avian reproductive technology at the University of Edinburgh. And now I'd like to hand it over to Dr. Rodley. The floor is yours.
All right, Michaela, you tell me if things are not as they should be
Yes,
in the slides.
everything looks good.
All right. Well, thank you for the introduction, Michaela. And I want to say thanks to SSR for interest in hosting a webinar on the topic of genetic engineering in food animals. What I'd like to speak about is the concept of engineering DNA blueprints for resiliency in livestock. And some of the work my program has been doing in this space, not just with research, but also thinking about the societal engagement aspect, as well as how we are trying to help to inform the regulatory landscape. At the most basic level, genetic engineering can be defined as the direct manipulation of an organism's genome. And this could be by way of human intervention. This could be sophisticated through tinkering with and altering DNA directly using gene editing methods, or conventional through reproduction using selective breeding, in which a genome carried by a sperm is combined with the genome present in an egg to now create a new genome with all of the elements that are necessary for life. And so through either the use of gene editing or selective breeding, human intervention engineers the genome of an animal. In modern times, we can observe the impacts of the ancient practice of selective breeding on livestock by appreciating the changes in phenotypes that occur over relatively short periods of time. So this image depicts the U.S. Grand Champion Shorthorn breed of cattle in 1835, which is the big one in the background. And then roughly 100 years later, in 1937, the Grand Champion of the same breed, and it was much smaller in stature. So just over 100 years of selective breeding dramatically changed the characteristics of the average animal. In the 1950s, the desirable cattle, and in this case, it's Angus breed of cattle, had a very small frame size. Only 30 years later, in the 1980s, the same Angus breed of cattle had dramatically increased in their frame size. And then roughly 40 years later, this is 2023, through selective breeding, the animal skeletons have begun to be reduced in size, and a sway was added into the spine. And so all of these dramatic changes in phenotype were brought about by selective breeding, and that in a basic sense is genetic engineering through human intervention. And so why humans work to change the characteristics of food animals, the answer to this is to address food security. This is an ancient interest, and it's one of the core reasons that livestock were domesticated. In high-income countries like the United States, food security is one of, I would say, one of the most unappreciated elements of society by most people, and that's because greater than 2 billion people on the planet are considered overweight. This represents roughly 35% of our world's population, and 90% of these individuals live inside high-income countries. But there's also nearly 1 billion people that are currently considered malnourished across the globe, and this represents roughly 15% of the world population, and nearly all of these people live outside of high-income countries. If we look at the number of deaths per year that is due to starvation or malnourishment, it's more than the deaths of cancer, malaria, foodborne illness, and HIV and AIDS combined. So there is a major global food challenge for not only addressing current issues of food security, but also the need to feed the world into the future. These data are the world population in billions of people from 1900 with a prediction to 2050 based on historic growth trends. Beginning in 1940, the world population started doubling every 40 years. So 1940 and 1980, it doubled in number and then doubled again by 2020. Based on these trends, there will be roughly 10 billion people on the earth by 2050. To feed 10 billion people at even just today's standards of amount and quality of food is going to require an increase of agricultural output by 60%. And this is just to maintain status quo and not even addressing current issues of food insecurity. So to address this pressing need, simply increasing agricultural output is unlikely to be the solution. This graph is plotting of arable cropland as well as grazing land that was not in use for production in 1950, 2000, and then a projection to 2050 based on historical trends. So think of these values as the percentage of arable land that's available for increased production. In 2050, roughly 97% of land that's amenable to crop production will be in use, and nearly 90% of all grazing land will be in use. So simply increasing production through land use is not a solution. And the future of food security is a global problem because we have decreasing agricultural land and increasing demand for food. So the world needs to find solutions for increasing the efficiency of both crop and animal source nutrients. And the demand for animal protein continues to grow. Between 1970 to 2000, it rose by nearly 80%. And between 2000 and 2020, demand increased by another 40%. This trend is predicted to continue as we move towards 10 billion people on Earth in 2050, and especially in countries that are moving from low to mid income and mid to high income. And so while there's surely many potential ways to address global food security problems, one primary solution is to try to enhance the efficiency of livestock production by engineering resiliency. What I mean by that is how well food-producing animals convert inputs, such as the amount and quality of water, the amount and quality of feedstuffs, as well as disease prevention strategies, into outputs for human consumption, such as milk, meat, and fiber. And of course, we're asking animals to accomplish this in an ever-changing environmental conditions, while also having less impact on the environment. And so the one intrinsic variable of the animal that influences the conversion of inputs to outputs and their resiliency is their genome. Selective breeding, being an ancient practice, has had dramatic and can have dramatic positive impact on genetic gain. But it's actually a messy process and it's not without challenges. First, selection for selective breeding is primarily based on what are deemed desirable or elite animals, and these are from observable phenotype, which may or may not reflect the merit of the genome. And selective breeding often takes 5+ generations or 10+ years to make incremental changes. And through this time, genetic drift, as well as inadvertent selection of negative phenotypes can occur. So the promise of gene editing is that we can combine molecular tools with curated DNA information to now devise strategies for efficient and precise genetic engineering to make significant improvements in traits within just 1 to 2 generations. Gene editing has been a revolution for many facets of biomedical and life sciences research, and this includes genetic engineering of food animals. Although there are several methods for creating gene edits in mammalian cells, CRISPRs are the most versatile, and they're the most easy to use, so have caught on as the primary tool. A key aspect of all of these gene editing approaches is that they can be applied without the use of foreign DNA or recombinant DNA to modify the genome, and they can bring about changes to DNA that can arise in nature. And so CRISPR has really become the key tool in the toolbox for food animal genomic engineers to efficiently and precisely genetically enhance food animals. I believe that the leading edge of applying CRISPR methodology in food-producing animals can be defined by 5 major areas. The first is improving welfare of animals, which is important for our holistic goal of improving the way we treat animals, but also the health of the animal for making food. The second is with disease resistance, which is critical to improve resiliency, reduce antibiotic use, mitigate zoonosis, as well as stop outbreaks that reduce food supply globally. The third is improving growth efficiency. That is how well the animals convert those inputs into making products like muscle. Fourth is environmental adaptation, which is resiliency and performance of animals in harsher climates, whether that be hotter or colder, or in environments with low resources. And the fifth is reproductive capacity, because transmitting genetic information to the next generation must occur through sperm and eggs. So enabling access to trait-driving genetics requires reproductive efficiency. The deployment of all gene editing applications that are currently being pursued for livestock trait development relies on efficient dissemination into livestock populations. And this has to happen via germline transmission. And so developing reproductive technologies can play an essential role in enabling access of embryos or gametes that are going to possess trait-driving genetics or the engineered genomes. So my program focuses on the male gamete as the route to widespread access and large-scale dissemination of desirable genetics, and that includes genetically engineered genomes. And that's because millions of sperm are produced daily through spermatogenesis. And so we believe the male germline is also amenable to direct germline gene editing and may provide an avenue by which we don't have to use embryos for editing. One of the most widely used technologies for achieving widespread dissemination of genetics from select individuals in livestock production has been artificial insemination. For example, we can isolate sperm from an elite male that exists anywhere in the world, put that sperm in a cryopreserved state, and we can ship it around the world for artificial deposit into the reproductive tract of females that are in the right stage of their reproductive cycle. So this technology has commercial origins in the 1940s and 50s and has had significant impact on dairy cattle production. However, there are limitations of application in other livestock production sectors. The tracking of reproductive cycles in females to know when to apply sperm and human skill for artificial insemination to introduce the sperm in the reproductive tract is a challenge. And so this works well in confined livestock operations where animals are observed every day. This doesn't work so well in range and pasture-based production in which animals are seen infrequently. And this is how the majority of beef cattle are managed globally. For swine, challenges with sperm survival during cryopreservation means that the reach of influence of an elite sire can have can be limited. In small ruminants like sheep and goats, artificial deposit of sperm in the female reproductive tract is technically challenging. So we believe that a solution to increasing access to sperm with trait-driving genetics is to engineer surrogate males that can produce genetically desirable sperm, and it can be disseminated via natural mating. So my research program has worked for many years to develop a strategy to achieve this, which we have termed surrogate sires. This is a CRISPR-based application to try to engineer enhanced reproductive capacity that is applicable to all livestock. I'm going to use cattle to portray the concept, but the applicability is essentially for all livestock. So you can imagine a multitude of different scenarios with various breeds of cattle or swine or small ruminants or really any mammal that could be plugged into this overarching scheme. So it starts with identification of a donor male. This could be one deemed to have elite or desirable genetics, or an indigenous breed male for any region of the world, or a male from something like a heritage breed. The next step is to isolate sperm-producing stem cells from testicular tissue for use as a fresh cell suspension, or cryopreserving the cells for later use, or placed in vitro conditions that support their expansion in number. The stem cells could then be harvested and transferred into the testicles of surrogate recipient males, in which donor-derived sperm production occurs, and by natural mating, the recipient's sire progeny with donor genetics. Two key aspects for this concept are the ability for in vitro expansion of isolated sperm-producing stem cells and the generation of recipient males that are ablated of endogenous germline so that all sperm possess donor genetics. I'm going to talk a little bit about the work we have done on this second aspect. So for roughly 20 years, several labs around the globe, including mine, have struggled with trying to address the question of how do you generate male livestock that don't make their own sperm, but still have functional testicles and could harbor regeneration of sperm production. So for most of that time span, using a genetic approach was not feasible because of lack of tools to effectively modify the genome of large animals. And then the game changer of CRISPR came along. And that enabled the ability for for the first time to effectively go after knocking out genes that are critical for only male germ cell survival, and in that way achieve germline ablation, but potentially preserve somatic support cell function. And so our approach has been to target genes that are crucial for survival of just male germ cells that, when inactivated in a homozygous state, will lead to male sterility, but female fertility would be intact. In addition, in a heterozygous state, both males and females would be fertile, and in this way, mating of females that are homozygous for an inactivated allele with males that are heterozygous for an inactivated allele will produce knockout sterile males, and we don't need to embryo edit to produce more sterile surrogate males. So we targeted the gene nanos2 because previous research in model organisms like flies that have a single nanos gene, genetic knockout results in male and female sterility due to germ cell death. In mice that, like all mammals, possess 3 nanos genes, targeted knockout using embryonic stem cell technology showed that knockout of nanos1 produced no phenotype. Knockout of nanos3 leads to both male and female sterility. But knockout of nanos2 led to male-specific sterility, but females were fertile. So we've worked to target nanos2 using CRISPR gene editing in a variety of mammals, including replicating the mouse findings, but also knockout of nanos2 in pigs, goats, and cattle. In all of these animals, nanos2 knockout males are germline ablated and sterile, but knockout females are fertile. And so over the last 8 years, we've worked on a proof of concept pipeline with several different species, the results of which are in published form, but also some are unpublished at this point. The pipeline starts with using CRISPR technology to generate nanos2 knockout animals and then phenotype them for germline ablation. If we determine there is male-specific germline ablation, but otherwise normal appearing testicular tissue, the next step has been to transplant donor sperm stem cells and ask, are donor-derived sperm produced? If the answer is yes, we then work to refine the process to achieve natural fertility. With mice, we've been able to check all of these boxes. With pigs and small ruminants, we've been able to check 4 of the boxes. And with cattle, we've been able to check all 5 boxes. Some of the evidence for this is in published form, and the rest is in process to be published, hopefully this year. These are some examples of the CRISPR nanos2 knockout livestock that we've created at Washington State University. And so we believe that surrogate sires has major application as a reproductive technology to impact livestock production, including swine and beef cattle in North America, beef cattle in South America, sheep production in the UK, cattle and small ruminant production in Africa, small ruminant production in India, and swine production. And so from my perspective, surrogate sires and other applications of gene editing for trait development are going to define what the future of food security looks like. Using these technologies, we're going to be able to achieve precision engineering of food animal genomes that can convert minimal inputs to maximal outputs and generate animals that are resilient and resistant so that they can be matched to their environments that we're asking them to thrive in. And we'll be able to do this in a climate smart way. So this grand objective is not without challenges, though. There's certainly a lot of science that still needs to be done, but there's also major challenges that are not directly related to science. And I want to spend just a couple of minutes talking about those, even though I'm probably over time at this point. In the livestock gene editing space, I believe there's 2 main challenges for achieving societal impact that go beyond research. And these are public acceptance and regulatory policies. With public acceptance, gene editing gets lumped in with a conventional GMO bin, even though the science and the intent and output are drastically different. As such, I think we need to find a way to shape a new narrative with the general public about what gene editing is and what we're intending to do with it. To achieve this, I think scientists need to work with bioethicists and communication specialists to devise holistic strategies to talk about our research in a way that is palatable, fact-based, and empathetic. With regulatory policies, most countries have statutes that were developed for the era of transgenesis and recombinant DNA technology. As such, they are antiquated and they're misaligned to the current science of gene editing. So I think there's major need for modernization and streamlining to create a minimal barrier landscape that is cost effective. And some countries are making progress in this area, whereas others are slow to move the needle. And so I believe harmonization and collaboration between the United States Department of Agriculture and the Food and Drug Administration of the United States is needed to restructure the regulatory framework. And if we can harmonize with what other countries, such as the UK and their DEFRA agency is doing, then I believe the outcome will ripple across the globe. And so with that, I will say thank you for the opportunity to present some of these concepts and ideas about where gene editing in livestock can go and what the future is going to look like. And I'm happy to pass the baton over to my friend and colleague, Dr. Mike.
You're good, Mike.
Am I good? Thank you.
Yeah.
Sorry. And you can hear me fine?
Yes.
Oh, so first, I'd like to thank the Society for the Study of Reproduction for the chance to speak to you today on gene editing in domesticated animals. And also, I'd like to thank my colleague, John Oatley, for giving such a great introduction and explaining all the concepts. But I will try to add a little bit to what he's already presented. So John works on mostly livestock animals, and my lab is really focused on poultry species and genome editing of poultry. So I just want to point out to you why chicken is such an important animal. And if we looked globally at the number of animals that were slaughtered yearly on the planet, the number that exceeds is chicken, with 70 billion chickens slaughtered per year. And if you imagine that there's now 8 billion people on the planet, that's over 8 chickens that are consumed per person on the planet. And as John also pointed out, our global scale of animal production is vast. Yearly, as I said, there are a huge number of billions of animals being slaughtered for consumption. And also there are different animal products that are being produced, 887 million tons of milk, and also again from poultry, 93 million tons of eggs produced yearly. And this demand for meat and animal products has soared over the recent decades, as you can see if we go from 1961 to 1920. And also, a large amount of this increase has been in Asia. And as John also said, we anticipate this is going to increase by 50% by the year 2050, owing to increased population growth. So all this production of these animals and animal products is having significant impacts on the climate of our planet, the environment, and also biodiversity. But we shouldn't frown upon this. We should also say that this animal production is important because it mitigates hunger, malnutrition, and poverty in low and middle income countries. Oops. So John mentioned selective breeding, and selective breeding has done huge amounts for animal production. Can I just ask, can you see my mouse?
Yes, we can see the mouse.
Okay, thank you. Thank you, I will use it then. We can see the improvement of starting with the broiler chicken in 1950. The broiler is bred for meat production, to the chicken of today in 2014. It has gone from taking 1 ton of feed to produce 330 kilograms of meat in 1950 to 1 ton of feed producing 590 kilograms of meat in the chicken of today. And this is an 80% improvement. And this is just by selective breeding, selecting the chicken with the best genetics that will be able to produce more meat for less food. And if you look at the amount of resources that would be needed to produce this feed for the chicken in 1950, if we use the genetics of 1950, if we had those on the planet instead of the chicken of today, we would need 80 million hectares of land to meet this global food production, whereas in 2014, we only need 45 million hectares. So we've almost doubled or reduced by half the amount of farmed land that we need for this production. So I want to address how genome editing technologies can help address future global poultry challenges. And we see that these tools can be used in multiple ways. They can increase the production of sustainable animal production to achieve global food security. They can help to mitigate some of these negative effects of animal agriculture on the planet through use of resources. Probably most importantly, they can reduce the burden of animal diseases, so the loss of production animals to disease. And in terms of poultry, we believe they can reduce zoonoses. So these are diseases that are carried by animals and can be transferred to humans. And this is very important for chicken and waterfowl. These tools can be used to combat antimicrobial resistance. And also, importantly, they can be used to enhance the welfare of farm animals and also conserving genetic diversity. And I'm going to spend the rest of my talk to address a few of these issues. So as John pointed out, I don't want to dwell on this too much. So we have these new genetic tools, which are called genome editors, and we use these to edit the the genomes of livestock. And simply, you can imagine that our DNA could be seen as letters in a book, and our genome makes up all the pages of this book that spells out the different genes or the different proteins that are made in our body. And these genome editors are so precise, we can go into this book of genomes and we can change one single letter in this whole book. And that's the precision. And we also have 3 different tools that we can use. We can use zinc finger nucleases, which are sort of the old type of genome editors that were developed 20 years ago. We have talons, which are also used, and again, they need 2 separate proteins to come together. And also we have CRISPRs that are our new tool. And these are guided by a protein, which is called Cas9, that cuts the genome at a particular locus, and a guide RNA that brings the CRISPR to cut the genome at any site. So birds are different than mammalian livestock species, and we actually don't inject these tools into zygotes, though most of us use a cell called the avian primordial germ cell for our genome edits. And I'd like to point this out just because I'm speaking to the Society for the Study of Reproduction. So I know that you'll be interested in this. So the germ cells, or the primordial germ cells, are the lineage restricted precursors to sperm or eggs of the adult animal. And they're present in the early embryo, and they're diploid, so they have 2 copies of each gene, and they're migratory, so they will move through the embryo. And what's interesting is that these cells are very difficult to culture. We're just learning to culture them for mouse and human, and also we're just beginning to be able to culture them for very short times for livestock species. But what's remarkable is that for chicken or for poultry, we're able to culture the primordial germ cells very well long-term in vitro, and they remain germ cell competent. So these are the cells that we're using to generate genome-edited poultry. And just to point out one more thing, because I know you might be interested, is that in bird species, the avian germ cells are preformed, or we think they're established by maternal determinants. So if you look in your laid egg, these primordial germ cells are present at these early stages. So we can take these germ cells out of an early chicken embryo from any breed of chicken, and we can culture them in vitro. And that's the point where you can genome edit them. So we simply sample an early embryo, we culture male primordial germ cells, and we culture female primordial germ cells. And then we can genome edit them with either our CRISPR-Cas9 or our TALEN tools. And then we introduce these back into surrogate hosts, and the surrogate host will carry these exogenous germ cells. And then when we breed them together, we would get pure offspring from these. But similar to what John mentioned in his talk, we've actually genetically engineered the surrogate hosts so they lack their endogenous primordial germ cells. So when we transfer our exogenous germ cells into our surrogate host, we can produce pure offspring from these germ cells. And this is just one example I want to show you. So the chicken on the right here is a white feathered chicken, and we simply took germ cells from that breed of chicken. We edited them to repair the gene that was needed to make feather color in this bird, and then we put the edited germ cells into our surrogate host. So in a single generation now in poultry, we're able to produce genome edited birds that are homozygous for any breed of chicken. So I'm going to follow on with what John was saying. I see that genome editing can have two major impacts on animal breeding. One of them is taking the rapid transfer of genetics between unproductive animals and transferring those to elite animals. So these are genes that might be important for disease resistance that have been lost in our elite animals. They can be now directly transferred into these animals without the associated drag or the genetics that would be remaining in these unproductive animals if we bred them directly. And also we can introduce new genetic changes that could create new beneficial traits in these animals. So I'm going to give you two examples of these, and I have to say, I found the first example this morning, and it's actually for a banana. It's not for a livestock animal, but I was just amazed that this progress had been done. And as you might know, if you eat bananas, we usually globally we eat a cultivar called the Cavendish banana, and we're in danger of losing all the Cavendish bananas on the planet because of a fungus that is infecting them, which is called the Panama disease fungus. And the Cavendish banana no longer contains the resistance gene, and this was lost during the breeding and selection to make it so productive. But what they did is they went back to the original banana that carried this which was resistance, and they re-isolated the gene that was present in this. So this was one of the original bananas, and they're not actually great to eat because of the large seed content. So this gene was taken from the original cultivar, introduced into the Cavendish banana, and it only varies by 9 amino acids during where it's found in the Cavendish banana where it doesn't work. And now the Cavendish banana is resistant to this fungus. So this is going to be very important for production going forward. I'm going to jump back to animals now for my second example. So this is a way we can introduce new genetic changes that could prevent a new beneficial trait in animals. And this is the PRRS pig. So this is the idea that we can introduce a genetic change that will introduce disease resistance. So PRRS is porcine reproductive and respiratory syndrome. It's a notifiable disease globally. It causes 1.5 billion in losses annually in the UK, and it's very prevalent in countries here. My last talk was in Ireland, so I pointed out that 50%, 56% of the flocks are positive in Ireland. This disease is very bad. It leads to abortion of the fetuses and also respiratory distress. So my colleagues at Roslin discovered they discovered that by removing a single exon in the cellular receptor that the PRRS virus binds to in cells, they could generate a pig that was completely resistant. So this is showing an unedited pigs here, post-challenge with the virus. And this is in the green line is the edited pigs. The virus could no longer get into the cells of the pig and able to infect them or to grow. So they're completely disease resistant. So what can I say? What can we offer you for poultry going forward? So I gave you one example, which was a banana, and one example, which was from a pig. So we think that genome editing can be used to improve poultry for both farmers and consumers. We think we can address three different things. We could increase disease resistance. We think we could increase productivity with lowering greenhouse gases. And one other thing we could address is to eliminate the hatching and culling of the wrong sex chickens. And I'm going to explain that in a little bit later on. So the first genome edited chickens that for disease resistance were done by my colleagues in Czechoslovakia. And a major virus that infects chickens is avian leukosis virus. And it binds to a specific receptor. So my colleagues went in and were able to edit this receptor, single receptor, and the virus was no longer being able to get into the cells, and the chickens were resistant to the virus. So I call this sort of a silver bullet trait. It takes a single genome edit to make the animal resistant. A bigger win for us and a more interesting target globally would be avian influenza resistance and being able to introduce this into poultry. And this, because avian influenza has such a huge effect on poultry globally, this would be a huge win for poultry and also for poultry across the planet. Just one slide to tell you about avian influenza, if you haven't heard about it, which I'm sure you have. So bird flu or avian influenza causes deadly disease in chickens. There's no effective treatment for it. We don't vaccinate for avian influenzas, and there's many reasons for that. So instead of vaccinating, we have to stamp out anytime avian influenza comes into our flocks to kill the flocks. And this has had huge effects in the UK. So between 2020 and 2023, we've had over 350 UK flocks with avian influenza where the birds on them had to be destroyed. And each year, because that's when we have, each winter when we have avian influenza come through, all the free-range birds have to be brought inside and kept captive inside, so they're not infected. So globally, this leads to huge economical burdens and health and welfare problems. And another fear about avian influenza is also zoonotic. It will spread from chicken to pigs, then to humans. And also there is a chance that it can jump directly to humans and set off a different pandemic. So we think genome editing chickens for avian influenza would be really key to prevent further infections in human down the line. So if we could actually come through and genome edit chicken for avian influenza, this would improve the welfare of chickens. So there's over 50 commercial breeds of chicken that are producing half of those 70 billion chickens that are culled for food each year. But also you should think there are 1,400 local breeds of chicken that exist on the planet that provide the other half of this food that's consumed by the global population. In that way, it'll help the farmers. These are both commercial farmers who keep these birds, and also local village farmers who keep chickens in their backyard. And also it'll help consumers globally. This is even high-income countries like here in the UK, but also low and middle income countries that keep a lot of poultry in village settings. So I will tell you one slide on our research, recent work. So we discovered a gene that avian influenza uses. So it's a gene that's present in chicken. And we edited this gene. We changed 2 amino acids in it. And what we showed was that in cells, that if you change these 2 amino acids, avian influenza is no longer able to grow in avian cells in vitro. So what we did is using our genome editing technology, we were able to introduce these 2 amino acids into these 2 amino acid changes into poultry. So when we took, in this case, a low pathogenic avian influenza, and we infected control chickens, the virus infected the chickens and it was able to proliferate in the controlled chickens. And when we had neighboring chickens or sentinel chickens in the same pen, they were infected with this virus. And when we did this with our genome edited chickens, and I'll just mention the name of the gene we edited, it's called ANP32A. The chickens, we introduced the virus into the chickens. The chickens were infected, but the virus did not grow, and the virus did not spread to sentinel chickens. So it was really a great result for our genome editing technology. So unfortunately, what we found is that the virus actually was able to rapidly mutate around this single edit in this single gene. And what we've discovered going forward is that we're actually going to have to edit multiple ANP genes in the chicken if we're going to introduce genetic resistance. So the third project for poultry that I wanted to point out is, and some research labs are bringing this forward, and this is called sex skewing of chickens. So if you eat eggs, you know that the eggs come from the female, and only female chickens are raised to produce these 90 million tons of eggs that are produced on the planet each year. And unfortunately, because of this, because the male chickens actually aren't selected for meat, they're actually culled each year, and they're culled at hatch. So in the UK each year, 29 million male chicks are hatched, and globally 6 billion female chicks are hatched. So the idea is to use genome editing to be able to selectively produce only female offspring, and that way we could actually breed female. We could only breed females. The male embryos would either not be ovulated, or they would not be, the male embryo would not develop. So in my final 2 minutes, which I'll try to finish in 2 minutes, so I'd like to just address the ethics of genome editing and also a bit of the regulation. So genome editing of livestock brings in new concerns as to the welfare of the farm animals. And a big problem is people who don't like seeing animals farmed for food now say that we're also going to use genome editing to actually keep this process going. And I would say the uses going forward of genome editing, I say that the big use, if we could actually increase welfare in farmed animals through making them disease resistant, I think this would be likely to be commercially accepted, not to increase yield, but actually increase their welfare by making them disease resistant. But I say that actually the first genome edited animal on the market is the red sea bream, and it was edited in Japan to make it have increased muscle mass with less food production. And also another ethical problem is the IP. Who owns the IP behind using these genome editing tools? And this probably will be a problem for low middle income countries, because we'd have to pay patenting issues or patents to be able to use these tools. And also, as John mentioned, the global regulatory landscape is a minefield. Each country has its own rules in terms of GMOs and also gene editing technology. We see a little change going forward. So in the UK, we have passed the Precision Breeding Bill. And what it says is that gene editing is in principle not different than conventional breeding. And it won't be, the animals will not be, crops and animals will not be edited or not be regulated as GMO. And also to allay fears about welfare, that the animals are going to have a welfare assessment to show that the welfare changed by the genome editing isn't detrimental. And also EU regulations, which are very prescriptive against the use of genome editing techniques in animals and plants, are being looked at again, especially for plants. And we might see some change going forward on the acceptance of genome edited plants and also potentially animals in Europe. And I'm going to finish there and just point out my funders who help fund some of the research I showed here. And I thank you, and I will stop sharing right now.
Thank you. Thank you, Mike. Thank you, John, for your great presentation. They were very interesting, and it's always a pleasure to learn your new results with your research. While I'm waiting for some question coming from the audience, I actually got some question for you. And the biggest is, what do you see the risk of, commercializing these gene editing animals? Well,
Mike, do you want to go?
Oh, I was going to say, can I have a break now? You can go first.
Oh, okay. Yeah. So I think the risk falls into two bins, at least in my experience in engaging with the public and engaging with federal regulators. It falls into two bins. One is the animal welfare aspect, which Mike touched on a little bit. And I think that this is almost self-regulating in the food animal world, because if we start with careful design and we do model organisms, say mice as a proof of concept, and then work our way up from there, we're already vetting for negative phenotypes or inadvertent phenotypes that come from the editing. And as a livestock developer or producer, if you have a welfare issue of the animal, that animal is not going to be thrifty or resilient, and therefore not a good production animal. And so that application would not be taken forward. So I already consider that self-regulating, that it's never going to make it to the commercial phase. The second thing is the safety for human consumption. And the big thing is that whether either the on-target edit or potentially off-target editing is going to create a new allergen by altering the composition of meat or milk. And I also think that one is almost impossible to address, because what defines normal in meat and milk? You can take genetically identical animals, feed them the same diet, but do it in different environments, and the composition of their meat and milk is going to be different. And just normal mutations that happen during DNA replication are also going to create potential allergens in the meat and milk. So I don't think that's actually addressable unless you were putting in recombinant DNA or something foreign into the genome. So I think those are the two biggest challenges and fears with commercialization: is negative impact on the welfare of the animal, and the impact on food safety. And the food safety is not about, for some people it is, that you're going to somehow inherit the genetic modification that's in the edible product of the animal. But the biggest concern is generating a new allergen in the edible product that's going to have a negative reaction.
Thank you, Trudy.
I'll add a comment to that. I think the biggest issue is consumer acceptance of these products. And you can imagine seeing if you went to buy poultry or eggs and it said, well, this came from, this is genome edited. But if if we could show in some way that actually it increases it has a benefit to that animal, I think that will be accepted by the consumer. And again, one of these things that's what we say avian influenza is so important for birds. If we could have, we're trying very hard to have some product that would be generated chicken that was resistant to avian influenza. I think right away that would be accepted by globally by consumers in many countries.
Yeah, I'll add that I think if we put labels on gene edited food, especially if it's food products coming from animals that have gene edits that could and do arise in nature, if you put a label on it, you almost kill it. It's just not going to get out, because the second that a label goes on something, people automatically, general public, I believe, automatically goes to there's something wrong with it. That's why you have to. And so I think that labeling could be something that really limits the commercialization and the actual deployment of genome editing applications out into food animals.
Yeah, I agree with that. And so, but like, how are we going to inform these consumers about the safety of this food? Because, I don't think everybody reads the scientific, literature, and even myself, sometimes I'm not up to date. So where are the format? How can we really make everybody aware of this genetic and animal safety?
It's not peer-reviewed scientific publications. It's communication strategies with the general public. It's getting into the grade school systems and giving young minds fact-based information so that when they grow up as adults, they're making decisions that are informed based on fact rather than fear or social media or other things. So we need to do better, I believe, in public communication and grade school education programs that are educating students, the future consumers of these products, about how they're created and why they're created. And I think that, some people still choose not to want to consume it, but at least they'll be doing it with fact-based information and not just as an emotional reaction. So I think science communication is absolutely critical. And we do not train basic researchers to do science communication. I have never trained a graduate student or a postdoc on how to do science communication. And so that's something that I think is missing in our training programs.
I was just going to add, we do have training. We do have training programs, and our postdocs and our students go and speak to the general public. So we do try to do that, and there are many publics you have to speak to, and of course you have to have an internet presence to be able to get your message out there. But the other important, consumers is great, but we also need to speak to farmers. So it's really nice to speak with farmers, small farmers, large farmers, and tell them about these animals. And a lot of the farmers are really supportive because they're the ones who see their animals die, and they're the ones who want the product, especially the PRRS pigs. They would love to be able to have some of these animals that are resistant to disease. I think it's important that the consumer sees that they know the impact of animals being lost to disease, and that needs to be communicated better and in more different ways. And as John said, not in scientific journals, because those don't matter. We're trying to convince ourselves of something, and we already know how this has benefits.
Yeah, I completely agree with you, Mike. I think the welfare is the key point to go forward, because with in this area where the awareness about welfare is so important, I think we can definitely have a voice. I have a question that came through the Q&A feature about avian influenza. And so, Mike, you said that the virus adapted quickly to the genome of the genetic chicken. Is there a concern that the edited genomes would cause a rise to several different strains of avian influenza?
Yeah, so the idea is that flu, if we can grow it in cells, we can generate mutations, and some of them could, spread more through cells or through animals, they'll have different degrees of virulence. So if I get the question right, what we saw in the chickens with the edit and how the virus changed didn't really frighten us that much. There were minor changes, and it changed the growth a little bit, but we were hesitant in that to actually. We reported that. That's why we challenged the system to see if we could cause flu to mutate and get around this change, because it was only when we gave a really, really high dose that we actually were able to actually be able to get flu that could change to get around this. So that is a concern, and that's why we don't think the single edited chicken can be brought forward or will be brought forward. So that was, but we know that because we know flu it comes back every year. It can mutate around almost any genetic change. So our idea is that we would have to go in and make multiple changes to stop the spread, to stop any mutation. And then we're going to have to test that in cells. And then again, we're going to have to test that in laboratory conditions to see if that's true. And I hope I answered the question.
Thank you, Mike. I have another question regarding the gene editing and how are we going to be able to maintain the genetic diversity if we're going to have a gene editing for a specific gene that is going to be adopted worldwide?
I think it needs to be introduced. So the gene edit, or whatever the application is, needs to be engineered into lots of different breeds and lots of individuals in those breeds. And this is where embryo editing may not be the course. That if we can figure out how to do direct germline editing, then we can just be editing specific males within populations and breeds, and therefore deploy it in a way that is not going to bottleneck the genetic diversity. So embryo editing, I don't think, is the future. I think direct germline editing has got to be the future. And they can already do it in chickens, so I'm jealous.
Well, we'd have to do this in cells. We'd have to do it in multiple cells from multiple individuals, because poultry is kept incredibly. The elite stocks are kept in large flocks containing 10,000 individuals. So, and they would not want these flocks to be bottlenecked. So it's almost the idea of, I'm glad I don't have to do this, and I'm glad I don't have to think about it. But then the idea of how many individuals you'd need to edit so you don't put the whole population through a bottleneck, it's going to be very difficult. Yeah.
Yeah, and I think there needs to be some kind of regulation if this is going to happen. And in my example, when I talk to students or people, is just we are regulating thoroughbred horses. We cannot use artificial insemination. We cannot use semen from all over the world to try to not bottleneck their gene diversity. And I think for gene editing animals, they will need to have something similar. And then I had a question, a curiosity for Mike, because you talked about so many different breeds of chickens. So if you need to choose a chicken that is going to go and be gene edited, how would you choose that breed?
Yeah. So that's what I was going to say. That's difficult because we have so many different breeds, and they're with 1,400 breeds. How are you going to do this? And that's what I say. I think we're a little bit limited by the intellectual property for the CRISPR tools and also the regulation. So this has to be done by a commercial company with a large commercial product. At the way the system is set up now, there's no way that you could go in with a small local breed in a low middle income country, edit that, and release it. It's just the funding is not there, and the costs that you would need for the process are prohibitive. So that's a big problem. And again, I like commercial chickens are nice, but I like the 1,400 different breeds that are, or types of chicken that are out there.
Makes sense. Thank you. And then maybe a last question for both of you. Which one do you see to be the next gene editing animal that is going to be commercialized, and when is it going to happen?
I would say so. This will be the PRRS-resistant pig. This was produced by Genus. It's going through the regulatory process in the U.S. now, and this was released. So this is by by the FDA, and that's expected to be out. Well, it could be out late this year, potentially, or next year. So it's very soon. That would be a PRRS-resistant pig.
Yep, I agree. In the US, it's got to go through FDA approval, like full FDA approval. And so we are not, nobody's privy other than the FDA to what products are actually in the FDA approval pipeline. And so we don't necessarily know. We can look to the literature and see what's being published to suggest what might be next. But even things, again, publishing in the peer-reviewed scientific literature is great because we just inform all the scientists. But that doesn't really get things out into the public domain in this case. You have to go through the FDA approval process and the commercialization process. And as scientists, academic scientists, we don't necessarily know what's in the big commercial entities' pipelines of development and who's actually in an FDA approval process right now.
Thank you so much. I think we ran out of time. It was a great opportunity to be the moderator. Thank you, Mike, for being here, and thank you, John. I think I will thank everybody for being here, and see you next time.
Thank you.
Thank you.
Wonderful. Thank you, everyone. And that will conclude our webinar for today.